Replication. This DNA is a molecule made up of two strands, and those two strands form a double helix. Each of the strands is made up of nitrogenous bases, that is thiamine, adenine, cytosine, and guanine. The two strands are complementary. Each strand has 5 end primer and 3 primer end, and those two strands will be running in two directions. It can either be from 5 to 3 or from 3 to 5, and these determine the strand which is being replicated. The first of which that determine this is to separate the two strands by the unzipping, which will be done by enzyme helicase, and this form a fork-like structure. So from that point, the separate strand each provide a template by creating a new strand, and this will be done by an enzyme called enzyme primase. The enzyme primase starts the process of which this enzyme makes small pieces of RNA called primer. This marks the starting point for the construction of the new DNA strand. The enzyme DNA polymerase binds to the primer and make new strand of DNA. This primase only adds DNA bases in one direction, either from 5' end to 3' end or 3' end to 5' end, but it will be doing from 5 to 3' end. One of the DNA now strand will form leading, which is the leading strand, and it's made continuously, and the second one is the lacking strand, cannot be made continuous way because it runs in opposite direction. The lacking strand cannot be made because it's what? Or opposite direction. Then only makes this strand makes of chunks called Osaki fragments. So we are saying what? The DNA polymerase only makes this strand in series of chunks called Okazaki fragments. You can check the Okazaki fragment. So from that point, each fragment is started with only RNA primer. So the DNA polymerase then adds a short row of DNA bases in a prime, that is 5' to the 3' direction. The next prime is then added further down the lacking strand. Another Okazaki fragment is then made, and the process is repeated and repeated and repeated. Once the new strand has been made, we will form a complete fragment. From that point now, we have another enzyme. That is, once the new DNA has been made, the enzyme exonuclease removes all the RNA primer from both DNA strands. And now from this point, the DNA polymerase then fills the gap and then are left behind with DNA. The enzyme DNA helicase seals the fragment of the DNA in both strands to form a continuous double strand. From that point now, the DNA replication is semi-conservative because each DNA molecule is made up of one old